Welcome to California Seaweed Festival Day 3, Seaweed Science. I'm Janet Kubler, coming to you from Southern California.
And I'm Simona Ogita, coming to you from Kailua Kona.
And today we have three great videos we're screening about science. Do you want me to introduce them, or do you want to go, Simona?
Sure, go ahead, Janet.
Okay, our first video features Dr. Kathy Ann Miller, who is the curator of seaweeds at the Jepson Herbarium at University of California, Berkeley. And it's a beautiful video put together by the public television channel in the Bay Area. I think you'll enjoy it because Kathy Ann Miller is the foremost expert on the biodiversity of seaweeds in California. Go ahead, Simona.
Yeah, and this is called Preserving the Forest of the Sea. So Kathy Ann will walk you through some of the seaweed specimens that they have, and also how they preserve these specimens at the herbarium. So you can learn about how they digitize and things like that. So I am going to share this video, so hope you enjoy.
Many people pay no attention to seaweed at all, even though they're stumbling over it on the beach. But seaweed is the forest of the sea, forming communities in which other plants, other animals live and thrive. Seaweeds are providing our oxygen atmosphere along with the land plants. Without these photosynthesizers, we would be literally gasping for air. My name is Kathy Ann Miller. I work at the University of California at Berkeley in their herbarium, where I'm a curator, or a person who takes care of seaweed specimens. An herbarium is really a plant museum, a treasure house of beautiful specimens that tell stories. Tell the story of the past, tell the story of the present, tell the story of the future. I'd like to introduce you to some friends of mine. Here are the green seaweeds, the brown seaweeds, and then we move into the red seaweeds. The reds have a place in my heart, I have to say, but the California seaweeds, those are my favorites. There are many, many species of seaweed in the world. There might be 10,000 species, and our university herbarium represents more than half of those. Seaweeds are just a subset of a greater group of organisms on Earth called algae. Algae occupy the ocean, freshwater, and land. Seaweeds are specialized. They only occur in the ocean. We're at China Beach, part of the Golden Gate National Recreation Area. This is Postelsia, a seaweed native to California, a brown seaweed, in fact, a kelp. This is the holdfast of the seaweed, specialized branches that holds the seaweed in place so it doesn't get washed away. This is the stipe, flexible, perfectly good for being bashed by the waves and standing up again. And then at the top, we have a cluster of blades, and these blades bear the reproductive structures. You can see how brown this is. This pigment allows photosynthesis to happen. So unlike plants on land, this baby photosynthesizes all over its body. Very, very efficient. Here at China Beach, you can see the intertidal zone. Creatures here are half the time out of the water, half the time in the water. They have special adaptations for this. They have compounds in their cell walls that help them retain water when they're in the air. Also notice how they're aggregated in groups. These groups helps them retain water. So this is one of the ways they can live in an extreme environment and feel like they're in the water all the time. Seaweeds of California are threatened in a lot of different ways. Climate change, of course. There are more and more non-native species, many of them invasive. So without really knowing what we have in our holdings, How can we apply the lessons of the past to understanding what we have today and where we're going in the future? Here we go. Ready for the first one of the morning? We were funded by the National Science Foundation to inventory our West Coast seaweeds, Oregon, Washington, California. 1 year later, we have taken high-resolution photos of 76,000 specimens. The next step is to database the collection information, including latitude and longitude of each collection site, so that specimens can be mapped. With global climate change in the ocean, I think we're going to find that southern seaweeds will be moving further north as warming happens. Each species will take its own course. Some will be enhanced by warm water, others will be completely extincted. Once we get the records together, the maps created, we'll be able to make predictions of what might be happening in the future. The students are able to process, to digitize 400 or more specimens a day. They're incredibly fragile, and the very fact of handling them so much was a huge concern to all of us. And I think we did a good job. Because I'm an expert on the native species of California, I've been called to help understand the non-native species, the invasive species that have come from other parts of the world, mostly through human transportation. Most of them are from Asia and may in fact be out competing some natives. It's very hard to tell them from the native species, even with 30 years of experience. DNA sequencing, genetic techniques, allow us to identify and clearly distinguish them from our native flora. So we've been developing a mystery here. A seaweed was collected in California that appears not to be a native. After the DNA extraction of our two samples, the native and the non-native, we've come up with the sequence here. This sequence matches exactly the sequence of Daisya sessilis from Japan. The second line is our native Californian Daisya senicola. Notice that it's related, but as you go along this way, they do not match. And that is the smoking gun that tells me our non-native seaweed is different from our native seaweed. So there's the puzzle and the mystery of it all. Trying to understand, is this new, is it non-native? But in addition to that, it's just the sheer glorious beauty. Their colors, their textures, their smells. It's just a fantastic assemblage of time and place. And maybe our future depends on creatures such as these.
That was beautiful, wasn't it?
Yes. Kathy Ann Miller's just done such an amazing job at the herbarium, curating all these species.
So next up, we have a video from Tom Bell at the University of California, Santa Barbara. UC Santa Barbara has been a hotspot of kelp science for 50 years at least. It is a place where people have really endeavored to understand the local kelp forests, from the ecology down to the individual organisms. So this video really shows some technological developments in kelp science.
Yeah, so you'll get a chance to see how they're mapping out the kelp forests in California, both above ground and also underwater. So we hope you enjoy.
Coming. My name is Lindell. Today I'll be telling you about the Scalable Aquaculture monitoring system.
I think we're having a little difficulty with the video.
Santa Barbara, where I am
Oh, here it is.
investigator.
There it is.
So thank you for coming. My name is Tom Bell, and today I'll be telling you about the Scalable Aquaculture monitoring system, which is a Department of Energy funded project at the University of California, Santa Barbara, where I am a lead investigator. And so our goal for this project is to design an autonomous monitoring system to monitor a kelp farm from outplant to harvest. And so this monitoring system is designed for offshore kelp farms, which one day might provide feedstock for biofuel production. And we use both aerial and underwater autonomous vehicles to monitor giant kelp forests like the one you're about to see in this video. And so we'll use both aerial quadcopter drones with various optical and thermal sensors, as well as autonomous underwater vehicles, or AUVs, to monitor the outplant success and growth and condition of kelp outplants on offshore farms. And because none of these offshore farms exist, we leverage natural kelp forest systems to develop techniques to estimate both kelp biomass and condition. And we test our underwater vehicles on a test farm located off Hope Ranch near Santa Barbara. And so the kelp forest you're seeing here is the Arroyo Camado kelp forest, which is located about 15 miles west of the UC Santa Barbara campus. And one of the things you can see immediately are the different colors of the kelp canopy. And these different colors represent fronds of different ages and physiological condition floating at the surface. And like I said before, we use aerial drones to monitor this canopy. We use both hyperspectral and multispectral sensors to monitor the condition of the canopy, and we can create maps of kelp biomass and kelp condition and the surrounding environment. So this is one of those maps we can create with the sensor you just saw on that drone. So here the different shades of green represent kelp canopy fraction, which is analogous to the canopy biomass. And the red to white to blue color shows sea surface temperature. So there's a creek that runs over the beach into the ocean at Arroyo Camado, and that red warm water lens is coming out of the creek and sitting on top of the cool ocean water. So when we look at the kelp canopy from above, like you do in these aerial videos, you can see that the biomass and condition of the canopy vary greatly across space. So it's important to validate the data collected from these aerial sensors with in-water measurements. And so we've partnered with the Santa Barbara Coastal Long-Term Ecological Research Project to provide diver estimates of kelp biomass, physiological condition, and nitrogen content. And here you can see a team of LTER divers about to don scuba gear and collect these in-water data. And we've also implemented our own monitoring experiments to track kelp through time and monitor changes in condition and sugar content. So here you're seeing one of our kelp tagging experiments at the Mohawk kelp forest near the Mesa Lane Steps. And so here we go out and tag growing kelp fronds every 2 weeks, and we return to those same fronds to sample blades and track the pigment content and sugar content of these blades through time. We also have a robust underwater monitoring project, and we do this now using remotely operated vehicles, or ROVs. And so this is ROV footage of the Mohawk kelp forest, and you can see the giant kelp attached to the rocky reef at the bottom and floating up to form the surface canopy at the top. And so we have been using both video and color stereo camera images from the ROV to monitor outplant success and outplant growth rate on our test farm near Hope Ranch. And so each one of these yellow tags shows an individual kelp outplant, which we are monitoring through time. And we're using deep learning techniques to automatically classify our color imagery to both size and determine the outplant success of each one of these kelp outplants each time we visit the farm. And while the ROVs have been extremely useful to develop our underwater monitoring techniques, we are now transitioning from using ROVs to autonomous underwater vehicles. And so we've partnered with the Navy to integrate our color stereo cameras and side scan sonar systems onto 3 lightweight and easy to produce AUVs. And so these AUVs consist of both 3D printed and easy to obtain parts and are completely autonomous. And so the line you're seeing attached to the AUV in this video is actually attached to a surface buoy just to track its movements from a boat on the surface. So these are able to navigate by themselves on preset tracks to record imagery underwater. So the vehicle can actually travel at a relatively high rate of speed, but we won't be approaching those top speeds to optimize our imagery. And looking inside the vehicle, you can see that there's 2 battery compartments which allow us to monitor for several hours at a time, as well as payload space for both our color cameras and side scan sonar. And in the nose cone, we can attach sensors to track changes in depth, seawater pressure, conductivity or salinity, as well as temperature, which is related to seawater nutrient conditions in our coastal upwelling system. And in these pool tests, you can see that the AUV is slightly positively buoyant, so if it ever stops for any reason, it'll float to the surface. And in the handle, it can transmit data through satellite or cell towers. And I want to finish the talk by showing you this shot that I really like of the kelp forest canopy from our aerial drones. And once again, you can see the different colors of the canopy, which are related to the pigment concentrations and sugar content of the canopy kelp forest biomass. And so we're using this information to try to optimize harvest time for these future offshore kelp farms. And so once again, I'd like to thank you for listening to the talk. And if you have any questions about the Scalable aquaculture monitoring system or how we can use these technologies to monitor natural kelp forests, I welcome any emails. So thank you very much.
Well, we hope you enjoyed that great video by Tom Bell and his colleagues at UCSB. I think it's fantastic that we have the technology there to monitor the kelp forests. We do also realize that there is a lot of native tribal knowledge from this area about what the kelp forests have looked like over the past. Over the last couple centuries. And we would like to highlight that in the future at our California Seaweed Festival. So I think integrating some of the old traditional knowledge with, some of these new engineering devices could be synergistic, So with that, yeah.
Oh, I see. We had another question I see also in the chat, which was asking if seaweeds grow in freshwater. And I just have to say that seaweeds have evolved to live in salt water, and very few seaweeds grow in freshwater. There's some grow in some kind of brackish, like lower salinity water, but not in freshwater. So there are algae that live in freshwater, but we wouldn't call them seaweeds. So, and also someone asked if we asked about growing kelp at home. And it's certainly possible to grow kelp at home, or I have grown smaller seaweeds at home. Kelp being so big, like you would need a really like commercial size place to grow it for any length of time. But you can easily grow some small seaweed in a small volume of seawater if you keep it clean and well lighted.
It will probably outgrow your aquarium pretty fast, though, especially if you're trying to grow giant kelp, because it grows at least a foot a day. So
yes, it's, you know, but it is a fun thing to think about doing.
For sure. You can definitely grow other smaller seaweeds at home, and there are kits available. You know, if you want to grow some microalgae or some smaller things in terrariums or aquariums, I guess.
Right.
So that's that's definitely available.
Okay, so you want to move on to our final video for today?
Sure.
Okay, our final video showcases a kelp breeding program in the North Atlantic, and that is not California, though some of the people working on that project are associated with the California Seaweed Festival and working in California. It shows you the biological technology evolution of techniques happening right now at a great pace in the US and in California.
Yeah, so some really great work, and this work that will be presented by Scott Lindell from Woods Hole, as well as Yukon. They're doing work with sugar kelp, but a lot of these techniques are similar for giant kelp, and there are groups working on those, and we will hear from a group today in our science panel at 4 p.m. So make sure to tune in for that. Okay, so here we go. This is our last video.
Hi, I'm Scott Lindell, Research Specialist at the Woods Hole Oceanographic Institution and Lead Principal Investigator for a sugar kelp breeding project funded by the U.S. Department of Energy's ARPA-E Mariner program. Today, I'm going to give you a tour of our breeding project and the supporting facilities involved. Here at the University of Connecticut and in Woods Hole, we have collections of wild kelp from the northeastern United States. These consist of hundreds of gametophytes from nearly 18 locations and tested at 2 research farms. For the past 2 years, our team has been using these to make hundreds of unique single-pair crosses of gametophytes each fall for grow out on research farms. Here's how the selective breeding process works. First, a quick reminder of the basic biology that is involved in kelp breeding. Every kelp blade starts from the fusion of eggs and sperm from female and male gametophytes, which are the microscopic alternative life stage that ultimately creates the blades or sporophytes. Our first task in a breeding program is to induce wild sporophytes to release spores that will become gametophytes. These are then separated mechanically into 96-well plates. Healthy, vigorous gametophytes are stepped up into ever larger vegetative or clonal cultures under red light. It can take up to a year to develop enough biomass for creating single-pair breeding crosses. About 6 to 8 weeks before we anticipate planting a farm, we induce our gametophytes to develop reproductive fertility by giving them an optimum environment. Then we introduce each unique male to a unique female gametophyte culture in order to create several hundred unique crosses. 2 weeks later, after they reproduce and develop sporophytes, we start the next stage of the nursery process. This requires applying the newly developed sporophytes from each cross to pucks, or pieces of PVC pipe that contain about a meter of wound seed string. We do this hundreds of times, so we eventually plant hundreds of 1-meter plots on the farms. The pucks are incubated in individual containers in a controlled environment, and after about 4 to 6 weeks, these pucks should look fuzzy, like a chia pet, with 1 to 2 millimeter sporophytes. That means they are ready to plant in the common garden test farms. Next, it's time to assemble the farm with the pucks and the grow lines. The trick to planting these lines lies in the preparatory work in the wet lab beforehand. There we carefully assemble the pucks into larger spools from which we can efficiently string consecutive plots onto the ropes. Then we unwind the string onto the grow ropes in the wet lab and place the ropes in coolers with seawater. At this stage, the puck strings are transformed to individual plots on the grow ropes. Then we move the coolers to the boat and prepare to reach the farm. Our common garden farm is an array of 5 parallel lines that are each 60 to 100 meters long and are closely spaced, just a meter apart. At the farm, the 5-line array is already set up. We simply attach each of the new grow lines, dump the cooler overboard, and then stretch the grow line out to attach the other end. Now it's time to sit back and wait. Our growing season is typically November to early June. Once late May or June arrives, we harvest the kelp before it starts to attract biofouling. We harvest each line, rope and all, and place it into insulated totes. Then these are trucked back to our lab in Woods Hole, where we can circulate 10 degrees C water through it overnight. The next day, we have staff working in pairs to evaluate every plot. First, we take measures of wet weight and blade density. Each 1 meter plot is hung up to weigh and then to snap a photo. Then subsamples are taken from the plot for enumeration of blade density and further analysis. Next, we complete the phenotyping of individual blades from the subsamples for each plot. The blades are laid side by side and measurements of each blade and stipe length and width and blade thickness are recorded. We record the condition of the blade, such as whether there are any fouling organisms or grazing, and whether the blades have source tissue. Each blade is individually weighed and subsequently dried in a greenhouse to record percent moisture. The dried tissues are analyzed for carbon and nitrogen, ash, sugars, and about 20 different ions and metals. So that concludes our tour. I hope you have a better understanding of the challenges involved in kelp breeding. Over the next year, we'll be publishing results of the important traits we've measured, many of which are highly heritable. We're very optimistic about the opportunities for improving kelp production through selective breeding, and we expect we'll meet our goal of achieving 10 to 20% improved yield per generation. I'm happy to take any questions now.
Of course, Scott Lindell is not here to answer questions now, though he did give us, you can contact him at Woods Hole if you do have questions. Also, we'll have in our panel this afternoon, we'll be having a representative of another kelp breeding program who can answer questions.
And also, you might have noticed in the video, I was doing some of the painting, so I was actually involved in this project before my current position at Ocean Era. And so if you have any questions, let us know in the chat box. Otherwise, feel free to tune in today when we have a great lineup of scientists telling us about all kinds of fun research that they're doing with seaweed in California right now.
Okay, thank you for joining us for the video screening on day 3. And
Thank you so much, and we'll see you in a few hours. Hope you can join us soon. And if you missed anything, feel free to check our YouTube channel. All these videos are being archived, so feel free to subscribe and follow us on social media as well.
all right. Okay.
Talk to you later. Bye-bye.